Hello everybody. In the context of the cancer community sessions, it's a pleasure for me today to present you a guest, a very dear guest that is a long-term collaboration partner of us, and it's Sabine Kasimir-Bauer from the University of Essen, and she will present us today her work in the context of what we call ELIMA study, which deals with the molecular analysis of circulating tumor cells, as well as extracellular vesicles and nucleic acids in liquid biopsies. Sabine Kasimir-Bauer is a professor at the University of Essen at the Department of Gynecology and Obstetrics. She is head of the laboratory, and she is a long-term researcher in the field of liquid biopsies. Sabine, I found an article that's very long time ago, probably it was 2002 or so, when you started with the disseminated tumor cells. Actually, is that true, 2002?
I started 1997.
1997. So you see what I mean. I mean, she is very long in that field now. It was in that days the word liquid biopsy was probably not there actually. So when we were talking about these disseminated tumor cells, that was followed then later by circulating tumor cells. But nobody could believe really, Sabine, that we end up here with what we know now about what's going on in cancer and what the modern research can provide now, correct?
Correct.
I'm really happy that Sabine today can present us a very comprehensive piece of work comparing all these analytes in liquid biopsy, and I'm really looking forward and I'm quite curious for your presentation. But unfortunately, before we can start with that presentation, we have to do a little legal things, so I have to put a disclaimer here to make sure that everybody out there in the auditory understands that we are talking about research. All the QIAGEN products that might be mentioned or so, it must be clear that this is not for a use in real life diagnostics or something like that, but research only. Having that said, Sabine, it's a pleasure to have you here. And the stage is yours now.
Thank you very much, Sigi, for the kind introduction. Well, hello everybody. It's a pleasure to present a part of our research here. As already indicated, I'm head of the laboratory in the Department of Gynecology and Obstetrics, and our main research fields are breast cancer and ovarian cancer. And within these fields, our main purpose is the detection as well as characterization of disseminated tumor cells in the bone marrow, called DTCs here. And we are studying CTCs, so circulating tumor cells, and other liquid analytes. So today's topic will be breast cancer and CTCs and other liquid analytes. So first of all, I would like to give a little clinical introduction, because this is very important when we think of using liquid biopsies later on for patient diagnostics or to look for predictive markers. So in breast cancer, if we think about therapeutic options, the target in primary breast cancer is still the primary tumor. So what is the problem? The therapy decisions are made according to the so-called predictive markers on the primary tumor. That means the hormonal receptors like estrogen and progesterone and the growth factor receptor HER2. But when the tumor has been removed by surgery, what we are really targeting is the so-called minimal residual disease, which is reflected by single cells like disseminated tumor cells and circulating tumor cells. And the question is, do we find the predictive markers that were proven on the primary tumor really on the single cells? And the answer is quite easy. No, the concordance between the expression of the predictive markers on the primary tumor and the single tumor cells is quite low, about 30%. So what is the main problem in breast cancer? We find a very, very early dissemination of tumor cells into secondary organs. As you see in that figure, The tumor consists of different cells, and some are able to leave the primary tumor. These cells move into the blood vessels. We call them circulating tumor cells. And from the blood vessels, these cells move into secondary organs like lung, liver, or the bone marrow. And these cells can leave the secondary organs, move into the blood vessels again. And it has been shown that these cells that move from the secondary organs into the blood vessels can go back to the primary tumor site, and these cells might be responsible for local relapse. So now everybody is aiming to study liquid biopsy because one other characteristic of breast cancer is that relapses can occur 10 to 20 years after first diagnosis, probably because of that early dissemination. So of course it's difficult for a medical doctor to analyze the bone marrow several times or take a biopsy of the liver or the lung. So everybody wants to use the blood as a real-time liquid biopsy. And we are not only talking about circulating tumor cells, but now we are also talking about cell-free DNA, about vesicles, about microRNAs, and others. So what is also very important when we talk about breast cancer is divided into different subtypes. And these subtypes are associated with different risks for relapse. For instance, the hormone receptor positive patients, this is the majority of patients, here shown in green, they have the best prognosis. However, late relapses occur frequently. Then we have the HER2 positive patients, so patients that express the growth factor receptor on the primary tumor. They can be treated accordingly. They have an intermediate risk. And patients at highest risk are the so-called triple negative breast cancer patients, shown in orange. They do not express any of the so-called predictive markers on the primary tumor. So what was the purpose of our study? One of our PhD students, Corinna Keup, who will talk in much more detail about this ongoing study, she did her PhD thesis in our lab. And she took care of hormone receptor positive patients. So what was the goal, or what were the tasks in her PhD thesis? So first of all, she should analyze CTCs, extracellular vesicles, and cell-free DNA from the same blood sample in one condensed workflow. And we wanted to minimize the blood volume needed. We wanted to minimize sample-to-sample bias, but we wanted to get the complete information about transcriptomic and genomic content. And these assays were performed in the follow-up of the disease. So what about the clinical study design? This is very important because you have to look for a quite homogeneous patient population, and this is very difficult in metastatic breast cancer. So what we did, we analyzed 35 hormone receptor positive, HER2 negative metastatic breast cancer patients, and our starting point was defined as patients who had either a relapse of breast cancer diagnosed years before and were to start therapy, or patients who had a documented progressive breast cancer before receiving a new endocrine chemo or experimental therapy. So finally, we analyzed the blood samples at this time point 0, but furthermore at 2 consecutive clinical staging time points, where also the medical doctors decide whether a patient has a progress or a stable disease or has responded to therapy. So, and if you want to correlate your findings with clinical response, you also have to stratify the patients. So in total, we had 105 blood samples, and we stratified the patients as follows. So at the beginning of our study, at time point 0, all patients, of course, were non-responders because they were progressive. But at time point 1, we stratified the patients into responders and into non-responders, and we did the same at time point 2. So at the end, we had overall responders on top, patients who responded all over the time, or we had overall non-responders, patients who had never responded to the given therapy. And we had patients in between who responded first but did not respond at the second time point, and vice versa. So what was the experimental study design? So this is now very complex and perhaps confusing at first view. I will go through this figure step by step. So first of all, what we wanted to do, we wanted to compare RNA signals in CTCs as well as in EVs. And the second goal was to analyze DNA, not only cell-free DNA, but also genomic DNA from the circulating tumor cells. So let's start with arm A and B. This was the first comparison studies, and the goal was to compare the mRNA transcripts enclosed in CTCs or EVs. We wanted to correlate overexpression signals with clinical outcome, and the question was, do the profiles differ with regard to the given therapy? So let me give you a short overview about the methods. This will be done in much more detail by Corinna Kolb in different webinars. So for the isolation of CTCs, we used positive immunomagnetic selection. We used beads that were coupled to 3 different antibodies. And for the isolation of EVs, we used membrane affinity-based isolation. We isolated the mRNA and performed qPCR assays. Let me show you the main results. This was a comparison of the transcripts enclosed in CTCs and EVs. So EVs shown here in dark gray and CTCs shown in black. So first of all, you can see that the profiles differ. The second result was that the transcripts involved in the PI3K-PIK pathway that was frequently overexpressed in CTCs. So if you look at the EVs, the main result was that the highest value was shown for a resistance marker, aurora kinase. And what is quite important, you remember that we are talking about HER2 negative patients, so patients who did not express the growth factor receptor on the tumor or on the metastatic tissue. But ERP2 and ERP3 coding for HER2, they were overexpressed in CTCs in about 33% of all patients. So, and this is another way to show how different the profiles were. This is a so-called Venn diagram, and you see that the overlap between the 2 analytes is very, very small. Now, what is also important to say, for those of you who would like to go into more detail, we are happy that these results were accepted for publication in Clinical Chemistry. This is an open access paper, and a podcast is also available. So what is also very important when we think of moving into the clinic, so use the liquid biopsy for clinical purposes. As I've already told you, we talk about different breast cancer subtypes, and we started to analyze these profiles also in another subgroup of breast cancer patients, the HER2 positive patients. And there are different results what we are talking about. So in black you see the HER2 positive cohort, and in gray you see the HER2 negative cohort, where I've already demonstrated the results. So what is different? So first of all, the CTC signals in HER2 positive patients are less heterogeneous. You see there are less black columns. On the other hand, mTOR and PIK3CA signals are common in both cohorts, and we also find a prevalence of ERP2 signals in CTCs, similar in both cohorts. However, in the HER2 positive patients, SRC, AKT2, and another resistance marker, ERCC1, signals significantly are more common. So, what can we conclude? So first of all, we were able to successfully analyze the first arms A and B, so we took care of the other arms C, D, and E. So first of all, we started to analyze cell-free DNA, and there were 2 different goals. So first of all, we had to establish a method to reliably analyze the cell-free DNA, and we wanted to use next-generation sequencing. So we didn't want to use our valuable cohort of 35 patients in the follow-up. So for establishment of the method, we used another cohort of patients with the same characteristics, also hormone receptor positive, but HER2 negative. And we started to analyze the cell-free DNA in these patients. We wanted to analyze the influence of the so-called unique molecular identifiers for variant verification. And we wanted to look for the prevalence and relevance of cell-free DNA variants. The second goal was, can we reliably analyze cell-free DNA after having depleted the CTCs, or do we have to take a separate tube of whole blood to analyze cell-free DNA? So first of all, what about the methods? I will not talk about the methods in detail. I will only show you one slide, which is, in my opinion, very important, the influence of unique molecular identifiers. So you see here the results of 10 patients where we analyzed PI3K hotspot mutations. So, and in black you see the allele frequencies with UMI analysis, and in gray you see allele frequency without UMI analysis. So one of the results was that a mean allele frequency above 3.5% and 4 EGFR variants were associated with worse overall survival. So 5 of the 10 PI3 hotspot mutations were verified with the UMIs, and 5 were not detected. So finally, we have to give a high DNA input and need good sequencing qualities that are necessary for variant verification. So this is one example with a high input of DNA. You see that the variant is detected. On the other hand, a low input might not detect this variant, and we might talk about a false positive result not using the UMIs. We successfully established the method, and these data are also published. This is also an open access journal, and all the details are presented in this publication. So the second question, can we reliably analyze cell-free DNA after CTC depletion, or do we have to take a separate tube of whole blood? The answer is short, yes, we can. So, and for those of you who want to go into detail, we are happy to say that these data are also published in an open access journal. You find all the detailed comparisons, and this shows you that you reliably can analyze the cell-free DNA after having depleted the CTCs without losing any kind of quality. So finally, we can say that we have established the method that we can skip one arm of our 5-arm study, and that we can reliably analyze cell-free DNA using next-generation sequencing. So now the whole, what is left is this arm C. This was really the most challenging arm. It took Corinna a long time to really reliably analyze genomic DNA from circulating tumor cells. So we can also put a green sign here. I will not talk about this arm. This will be detailed in Corinna's talk. The only thing I will show you is that we were able to analyze 28 of our 35 original cohort patients at time point 0. And this is a simple figure showing you at first view. So in blue, cell-free DNA sequencing, in red, CTCG DNA sequencing. And you see that it's much more complicated to analyze the genomic DNA in the circulating tumor cells as compared to DNA, but it works. And we are happy now that we in total can now analyze the whole cohort. The final analysis of the 35 patients in the follow-up of the disease is ongoing, And it's very exciting later on to see which analyte or which combination will be the most useful one to guide therapy decisions, hopefully in the future.
Thank you, Sabine, very much for this comprehensive introduction into the ELIMA study. And I cannot, or probably already we cannot wait for hearing Corinna's explanations in depth about how this all was done and what the results have been in detail. Let me now introduce a few more people here. This is going to be a little expert round for the discussion of what you were telling us, and in this purpose I would like to introduce Markus Sprenger-Haussels. He is one of the initiators of these studies from our side, being the senior director of the sample preparation R&D group here. And he will give us probably kind of an insight about what's ongoing in the communities in cancer research. And I'm also happy to introduce here Constanze Kindler. She comes from our marketing department, being a global product manager, and probably she can give us a little feeling about what the field is saying and where this is all going to in terms of liquid biopsy. First of all, Sabine again. I think looking back, so what we did in the past and what we see now, I think there is a new paradigm opening really, and your results clearly show that we are on a way to learn so much more and deeper about how cancer goes, how tumor biology goes. And this is unbelievable if we think about 20 years ago. So nevertheless, what do you think, where this is all heading to? What is the value, or is there a value of multimodality compared to single component analysis? And I also could imagine that you will not stop at this point. So what would be your focus for the near future?
Well, first of all, I think that we have to analyze different components of the blood samples, because we cannot only rely on one marker. The problem is that for each analyte we do not have a standard method. So there are so many methods on the market, and comparison studies are very difficult. So first of all, you have to evaluate your marker very carefully, and you do not know which marker might be the best one to indicate whether a patient will progress or will have a relapse of the disease. And so having more than one marker, so at least 2 or 3 markers in the follow-up of the disease. And of course, we have to enhance the cohort of our patients to really see which of the markers will be the most suitable one to help us in the clinic to detect a progress earlier and to make interventional studies, for instance.
Yeah, I think that's very true, and that brings me already to Constanze. I think you have the overview about what is going in the world. So is Sabine's work and her opinion about that fitting into the community mind?
Yes, definitely. Very often when I'm at conferences, especially liquid biopsy conferences, we do get a lot of questions, or we did get in the past a lot of questions regarding, okay, liquid biopsy, there are different analytes, what are the different starting materials, what is the best analyte to look at? There it was always discussed whether it makes sense to only look at one analyte and or to really look at different. And I think Sabine's presentation today is a very nice example for showing like the mRNA profiles in CTCs and EVs. I know that I have discussed with a couple of people before that, and said, so it would be of interest to look at this to see if there is really, because until today nobody could really answer that. And that is what I'm also seeing now in the community, in the market, that people start looking also at different analytes, not only one, but a couple of different analytes.
Yeah, I think that's really the way where this is going to. Cancer seems to be kind of, if you ask me, cancer seems to be like of a mosaic. It is not one thing, and it's never clear what is the driver, what to look for. But if you have the overall picture, then you know better. At least in the research, you know better where will that go to and what will happen. And let's see if we one day can transport that and translate that into something that really helps the patient. Markus, you are already smiling because of, I know actually that, and you know that in the beginning, and you have been the driver of this ELIMA study, this 5-arm study, where I was always suspicious if this is really, if we can make that in a 3-year event. And it's unbelievable that we made it happen. And what is now what now would you say? Did you expect that really, that we would end up like this, or would you have been happy also with half of it?
I didn't expect it, to be honest. I was
I thought
surprised
so.
by the outcome. First of all, many thanks to Corinna and Sabine, who did really a fantastic job to put such a complex study together in time. So why I didn't expect such an outcome, because the original driver for me to think and to support such a study is that we had so many different technical options in the field of liquid biopsy, but I haven't seen in the literature a clear technology comparison done systematically with a good outcome, and that's why I really wanted to see such data from one very well controlled cohort. So maybe let me summarize. We have the cfDNA, which I think is the longest liquid biopsy arm in the field. And here we have different options like technical, like NGS or digital PCR or PCR. It's, I think, well established. The problem here is that you always have a big background of wild type, and you look for mutations, for insertions, deletions. It can be a very sensitive method because if you have a zero background of mutation, you just need to find the needle in the haystack. On the other hand, you don't know where the tumor is coming from, and you always have you're fighting sensitivity. On the other hand, we have exosomes and mainly RNA profiles. We look into exosomes. This is, I think, a very early liquid biopsy arm. And here we look at a completely different picture. We look at transcription. So what happens in a tumor cell? Are their genes upregulated or downregulated? But we have a similar issue with background noise, because in the exosome population we see lots of signals coming from healthy cells, and the needle here is the exosomes coming from tumor cells. And then last but not least, the circulating tumor cells, which are also for quite a while in the market, but in my eyes, the focus for this field was, I wouldn't say wrong, but it wouldn't use the strengths of this arm, because many people just stain CTCs and count them. But I think the real treasure in CTCs is looking into transcriptional profiles, and at the same time you have access to the genomic profile. So it combines basically the cfDNA and exosome potential. But on top of that, based on the technology, we have a strong enrichment of signals, so we get a much better signal to noise ratio. And what was driving me now to compare these three methods in a study to really see is the theoretical strengths of one or the other, or the weakness. Can we see this in a study? My naive expectation was, if we see, for example, a transcriptional profile in exosomes coming from CTCs, and we enrich the CTCs and look into the CTC profile, my naive expectation was to see we're seeing a much better signal to noise ratio in CTCs, but we see the same signal in exosomes. And since CTCs are usually found at a later stage in cancer progression, maybe exosomes detect something earlier, but in CTCs you have a much better signal-to-noise ratio. And that was a surprise actually of the study, showing that the signals are not the same. We see almost non-overlapping. There is some overlap, but it was surprisingly low. And if you think about it biologically, it makes sense, because also the tumor surrounding cells have a reaction to the tumor, and you find maybe also responses here, plus cells which are not yet circulating in the bloodstream, still they start already excretion of exosomes. So that was the real surprise of the study. And I think the beauty of it, and that was the on top, I think, benefit outcome of the study, is that when we look at combining now these different signals, that we get a much better predictive and prognostic answer than looking just at single analytes. So my expectation was finding maybe that one is better than the other, but the outcome was, well, you have to look at everything to get really a clear picture of the complexity of a tumor event.
Sabine, if you talk now to your clinical colleagues and probably also in the compared to the scientific colleagues on the conferences, what do they think about it? For the clinicians, it must be something like, wow, there is something that may predict that I'm doing something wrong all the time. So how do they deal with that?
So this is not so easy to answer. On the one hand, all the medical doctors are permanently looking for new therapeutic options. But on the other hand, they have to follow strict guidelines when they treat patients. So I remember very well when I started at the end of the 90s with the disseminated tumor cells, and one medical doctor approached me and said, well, it's very interesting what you are doing, and I trust in your work, but what shall I tell a patient who has a small tumor, no lymph node involvement, good prognosis, but tumor cells in the bone marrow? You have to offer something to the patient. So, and what is very important, first of all, you have to convince the medical doctors that you have a reliable method. Second part is you have to convince them that what you are doing is of prognostic relevance. And secondly, the medical doctor has to use this result for a therapeutic decision. And this is a little bit difficult. I think the good news is that so many people moved into the field of liquid biopsy, and that we have so many clinical studies, for instance, on circulating tumor cells, and that now everybody is flashed more or less by the cell-free DNA. And I think that the medical doctors are convinced more and more because so many people jumped into the field. We have comparison studies. And for instance, for the circulating DNA, clinical studies have proven that patients who have PI3K mutations in their blood samples, that they benefit from a combination therapy of, for instance, PI3K inhibitor and a anti-hormonal treatment. I think our medical doctors are very convinced about what we are doing because, as you already said, we have to say that they really support the study, that they really call us and say, well, I have a new patient that might be interesting for your study. So they supply us with all the blood samples in the follow-up. And I think since so many people have jumped into the field and have proven that we are doing reliable work, I think the field is now moving much faster. That I think, for instance, DNA will move into the clinic at first.
That's an important last sentence. So because of who will make the race, and again the discussion is if this race is about one winner or are all necessary. Markus, you are involved so heavily in all these international consortia and where people try to find out, actually supported by governments, try to find out what is it now, what are we looking for. Could you give us a little overview about how these consortia work and what they are looking for?
Sure, yeah. I think this is really important, that it's not only single sites working on islands of technical solutions. So we are very active in two consortia I would like to mention. One is Cancer ID. It's an IMI funded consortium internationally with 38 partners from academia, clinical universities, biotech, pharma, so really from different disciplines. And then we have SPEDIA4P. It's also a large consortium, but they have both different focus. Maybe let's start with clinical with Cancer ID. And Cancer ID, here we actually looked in the first phase. We did a real method comparison. So for the different analytes like cfDNA, CTCs, exosomes, microRNA, we were first trying to compare different technologies. And so we were actually using all the technologies which were used in the consortium, sending centrally samples to all these sites to compare actually, do all these different workflows for the same analyte give the same profile and the same answer. And there we already got some surprising answers. I think in the field of cfDNA, we found quite a bit of a match, but we found still clear methods and workflows which work better than others. For microRNA, we found, for example, a much more differentiated picture that the kind of method you're using really has an impact on the profile. And then in the second ring trial, where we were now fixing the pre-analytical part, we were also comparing the back-end technologies to come up at the end with a recommendation by Cancer ID, what are sample to insight workflows which really provide the most robust and most trustful results. So that's, I think, was the biggest value of the Cancer ID consortium.
And this is published already, Markus, right?
It's partly published. The miRNA ring study has been published at Clinical Chemistry. The cfDNA ring study is approved, so it's going to be published, I think, end of September. And the second miRNA ring study is still in the final writing phase. And there's more to come.
Great.
So maybe two more sentences to SPEDIA4P. Again, it's an international consortium working on putting workflows together. Here the focus is a lot of putting efforts into ending up with standard operating procedures, SOPs, which finally end up in CEN or ISO guidelines, which really help the clinicians to give them guidance what they should do and what they shouldn't do. And so there is basically putting this into a regulatory framework also, and help translating the great research everybody's doing into clinical routine, what Sabine just mentioned. This must be the ultimate goal, that at the end the patients are making benefits from all these great findings.
Fantastic, thank you, Markus. So, and that brings me finally to back to Constanze again. So we heard that it's kind of getting people are getting more and more convinced that things are important, that liquid biopsy is important. Is that a picture you could also observe in the market? Is this fashion growing in research?
Yes, definitely. This is growing very strongly also. But from my perspective, I would say we are still at the beginning of liquid biopsy. Liquid biopsy, I think, has great potential. And like Sabine mentioned, now we are moving a little bit faster forward. Like the medical doctors are now more seeing the benefits on liquid biopsy. More people are joining the field. But I think there are still a lot of challenges that have to be addressed and overcome. Like Markus nicely explained and pointed out, there are the difficult technical hurdles, whether you look at cell-free DNA, CTCs, or exosomes. And we also heard, and can confirm that that here for the cell-free DNA we are closest to the clinic, and that is progressing very well. But I think especially for exosomes, there's still a lot that need to be investigated. And on the other hand, I mean, there's a reason why these consortia have built and why we have them in the field. There's a strong interest to push liquid biopsy forward, as we can all see. But what that also shows me, and also the study that we have done together with the University of Essen, with Sabine's group, is that we are now not looking at a single analyte, but at multiple analytes, and we already see and learn how this is pushing or improving the speed of getting liquid biopsy into an active application in the field. And again, liquid biopsy, I think, is at the beginning, and I think there's much more to come. Today we talked a lot about therapy or therapy guidance, but there's more. There's monitoring, and there's early cancer detection. I think that is also a very interesting part where liquid biopsy could play a big role and has a lot of potential still to come.
Thanks, Constanze. And that brings me to my last question, actually. So it has nothing to do with your study, but probably it has indeed something to do with the future of your work coming from this study. What do you think about the immuno-oncology?
Yes, this will make it much more complicated, but I think we have to include immuno-oncology. I did my PhD in the field of immunology, and I remember well that I ended up with interleukin 15. I don't know which is the highest interleukin now. I expect 45 or so. And this is also a very complex network. And if you think of melanoma especially, we have the checkpoint inhibitors that are very, very successful in treating the disease. And now there are also ongoing studies in breast cancer, in ovarian cancer. And I think now that we have drugs available, we are coming back to the roots and starting to also include immunology. I think there was one publication even in 1994 showing that those breast cancer patients that have a high infiltration of tumor-infiltrating lymphocytes have the best outcome. But during that time, no drug was available. But now, since we have the drugs, everybody is including the checkpoint inhibitors now into the clinical studies in every tumor entity. And we will see how this will work and what kind of patients will benefit from these therapies. But I think we also have to include this aspect in our multimodality studies.
I think so either. So thank you very much again to everybody, and see you hopefully later or hear you later in our upcoming webinars about the ELIMA study. Thank you very much.
QIAGEN.
Sample to insight.